So thank you guys so much for having me here today. And I'm so grateful to each and every one of you for all of your effort to raise funds and awareness for Alzheimer's disease. And so today I'll talk to you about parasite, this isn't working, Caldesmin and Alzheimer's disease. The first thing is, there's this mass gross pathology in the Alzheimer's disease brain that you can see in this first image. And then I became very interested in the vasculature of the brain. So blood vessels, what happens to our blood vessels in the brain as we get older and what happens in Alzheimer's disease. And as a postdoc, I got really interested in these cells called pericytes, not pericyte, that I'll talk to you about. And then, now I'm like, okay, if we know there's a problem with these pericytes, what causes that problem? Ta-da. So we know pathological hallmarks of Alzheimer's disease include A-beta plaques and tau tangles. You heard some about A-beta from the last speaker, but we've known about these pathologies over 100 years from Alois Alzheimer himself, who first described the disease. He also described neurovascular problems, so problems with the blood vessels in the brain. And for the most part, over the last many years, people have been very focused on A-beta and tau, but less about what is happening to blood vessels. And why your blood vessels matter is because that's how you get glucose and oxygen to your brain. If you don't have adequate nutrients to your brain, it could cause problems with functioning. And so the blood vessels in your brain are very organized in a specific way with big vessels at the surface. This is a cartoon I drew when I was a postdoc in PowerPoint. I think I have this thing so backwards. Okay, so you can see big vessels at the surface, so those are arteries. And as arteries dive into your brain, they become smaller and smaller. And the smallest blood vessels are called capillaries. And the cells that support those capillaries are these pericytes that you can see here. And then as the blood vessels return back to your brain, they become arterioles and arteries and go pick up more oxygen. And so one fancy tool that we have in my lab and one technique that I learned as a postdoc is how to do intravital microscopy. So we're using animal models. We can remove a piece of the skull and image into the brain. And the reason this matters is we need an intact system with blood flow if we're trying to understand what's going wrong with blood flow in the brain. And so what you're looking at in this video was red blood cells, or the little black things flowing through. So you can see those flowing. So we put a dye in the vasculature. Those are red blood cells flowing through the brain. And we really care about these small blood vessels, capillaries. And you can see the red blood cells go single file line through those capillaries. And then the little pink thing you see is the cell body of the pericyte, these cells that we care about. And so this is an example of one pericyte on the vasculature. Most all of your vasculature in your brain is supported by these cells, but this is a specific technique to be able to show you one cell at the time. So the pink is a pericyte. You can see it's wrapping around those capillaries in the brain. And so what do these cells do? They're very important for maintaining the blood-brain barrier, so keeping things from getting into your brain that don't belong there. And we know in Alzheimer's disease that the blood-brain barrier breaks down. They're also important for angiogenesis, the formation of new blood vessels. And we know in Alzheimer's disease there's aberrant angiogenesis. They're also involved in phagocytosis. So similar to the last talk, pericytes can take up A beta and also take up tau. And we know that this is disrupted in Alzheimer's disease. They're also important for the regulation of blood flow. So in Alzheimer's disease, we know there's a reduction in blood flow in your brain. And what is the cause of this? Nobody really knows. They're involved in neuroinflammation, and we know there's aberrant leukocyte entry, so damage, so this entry from your blood to the brain of leukocytes. And they also have kind of a fascinating stem cell activity. So it's been proposed that these cells can leave the blood vessel and become microglia like you saw in the last talk. So that's kind of a fascinating understudied area of this cell type. And so when I was a postdoc, I used a very complicated technique that hopefully I can explain in a way that you guys can get what I did. So there's a tool called optogenetics. So algae, you guys know algae in a pond, have a specific kind of channel that's light sensitive. So we took this channel out of algae, not me per se, but scientists discovered this tool that's light sensitive that we could genetically modify a mouse to put that channel into pericytes. And when you stimulate that channel with a specific wavelength of light, it allows calcium to come into the cell. So at the time, it had been debated, do these cells, are they involved in contraction and blood flow or not? And so this is data from that paper. So the pink thing again here is a pericyte. The white is the blood vessel underlying it. And what you can appreciate from this video is when we stimulate those cells with light, it causes a reduction of blood flow. Tiffany, can you play this one? So this, the light blue line that you see is the diameter of the vessel before we excited that. And you can see that, unlike the video I showed you earlier that had these red blood cells flowing through, when we cause that pericyte to contract and clamp down on the vessel, it disrupts blood flow. And so we also studied mice that have a reduction in the number of pericytes and found those mice have reduced cerebral blood flow, reduced brain oxygenation, impaired metabolism, and when they're old, they have neurodegeneration. So these are all similarities to Alzheimer's disease, so that's why I'm very interested in this cell type. A beta that you guys have been hearing about can also cause pericytes to contract and reduce blood flow. So here's another video. Tiffany, if you'll play that. So at the beginning, you'll see these red blood cells going through. And if we either wait or fast forward the video along, you'll see that there's this one pericyte here. You can see that pink. We just fast forward it a little bit. So you can see that when we put A beta on, it causes those red blood cells to stall out. So it directly is impacting blood flow. And this happened for minutes. I was like, wow, holy cow, this is amazing and really important. Because if you don't get proper blood flow, you're not getting the oxygen and glucose that you need. And so my postdoc lab is not the only lab that has found these things. Several other labs have also shown that pericytes are important for contraction and blood flow regulation, and that this A beta can cause them to contract. So in my postdoc lab, we also were involved in human clinical research. We know the blood-brain barrier breaks down with aging. It's worse with mild cognitive impairment. It seems to be dependent on this ApoE4 gene. And in the work of the Zlokovic lab, it seems independent of A beta and tau. But what we know kind of in summary about vascular problems is, When you're cognitively normal, your vessels are fine, but preclinical mild cognitive impairment, we start to see problems with the blood vessels and all the supporting cells. And that gets worse in early AD and as the disease progresses, but this is an earlier problem than A beta and tau. So for me, it's all about what is the root problem and how can we fix it. And so I'm interested to understand in Alzheimer's disease what causes reduced cerebral blood flow and trying to find new candidates for pericyte contraction that could be involved in reducing blood flow and aging in AD, and pick this protein tau desmin. And I'll tell you why in just a second. But we know that there's a reduction in tau desmin in Alzheimer's disease at the gene level, and it also seems like at the protein level based on our preliminary data, and that could ultimately cause many of the problems that we see in Alzheimer's, including this reduced cerebral blood flow, neurovascular coupling dysfunction, reduction in pericyte coverage, because in stroke, when these pericytes clamp down, they can go into almost like a rigor mortis state where they clamp so hard that those cells die. So it's possible that if they just get too contracted, that could lead to their loss. And also we know that if they're lost, it could cause these blood-brain barrier problems. So this tau desmin protein that we're interested in actually inhibits contraction. So you may remember from biology about actin and myosin. So you can see those here are really important for a muscle to contract. And this tau desmin sits in the middle between actin and myosin. So if you lose that tau desmin, you can have more contraction. So we found that tau desmin is highly expressed in pericytes in the human brain. So that's what you see here in this purple color. Col4A1 is another pericyte marker. UEA stands for the underlying vessel, but tau desmin is highly abundant in pericytes. And we also see it's highly expressed in mice pericytes. And this is the study I was telling you about. So until 2022, I think is the year this Nature paper was published by Tony Wisgray's lab, they were the first to do RNA-seq, this RNA sequencing on vascular cells from an Alzheimer's disease brain. And they published a wealth of data, all the data they got. So you can mine that to figure out which genes are changing in the Alzheimer's disease brain. And for me, I cared about pericyte. So, okay, which gene is changing in pericytes in Alzheimer's disease that may be involved with this blood flow problem? And that's how we found this tau desmin. So you can see that the control tau desmin levels are much higher than the AD levels in pericytes. And when we did some staining on brain tissue we had in the lab from people who unfortunately passed with Alzheimer's disease, we could see less of this protein in the pericytes in the brain. So we want to do more of this to know if that's true. And functionally, we did some cell culture experiments in primary mouse pericytes. So if we silence tau desmin, this protein, it makes the cells contract faster. So it really does seem to be working like we would have expected. So then this brings us to the aims of my CART grant. The first is that tau desmin is reduced in pericytes of mild cognitive impairment and Alzheimer's disease in the cortex and hippocampus. And that's compared to brains of people who had no cognitive impairment. And we predict that ApoE4 carriers will have a more dramatic effect than non-E4 carriers. And so when I was a graduate student actually at UAB, we received a cohort of tissue from the Duke University Brain Bank And my PhD mentor passed that to me. So we'll use this very well characterized human tissue to answer this question. And then the second is functionally what happens. So we want to make a mouse that has reduced caldesmin and pericytes and determine the functional consequence of that on both pericyte contractility and also blood-brain barrier integrity. This is just some of the methodologies, so we can isolate blood vessels from tissue as we need to be able to look at levels of caldesmin. And we also do this multi-photon imaging like I showed you earlier, where we can really image the live dynamics of what's happening to blood flow and to pericytes. And we also have a technique called laser speckle. So this lets us look at brain oxygenation. And so that's it for my presentation. This is my lab group. We have quite a few collaborators at South Alabama and beyond. I was very fortunate to found the Gulf Coast Alzheimer's Disease Research Center in the last year, starting to try to help going on, and that happened because Charlie DeCarli at UC Davis gave us a grant, sub-award of his grant to begin, enrolling people in South Alabama and looking at vascular problems in their blood and by imaging. And so thankful to CART, NIH, and I also have funding in the past from All's Out, which I can connect you guys with. It's just a group of young professionals in LA that I actually used to volunteer with that would work on raising funds. But similar to CART, it's a group that goes out, raises money, and gives 100% to research. So they're a great group. My money from that actually came from Johnny Weir, that's a figure skater going on Family Feud. So it's kind of interesting. But yeah, so happy to answer any questions you have, and thank you again so much.